Okay, sorry I can't be there. I am a postdoc at Georgia Tech and I have research obligations. But I was able to pry myself away from my busy schedule for a 15-minute warm-up talk, which probably won't take 15 minutes. So I'm Samantha, for those of you who don't know me, and I am predominantly a microbiologist, but today I'm going to be trying to describe to you very basic general concepts in molecular biology, which lead up to understanding some of the researchers that are talking about prebiotic chemistry. So for all you non-biologists, this one is for you, and I will try to keep it simple. So life, what is it all about on this planet? Well, from a bacteriologist point of view, such as myself, focusing on therefore bacteria, life produces a number of things for us, such as oxygen. I like to breathe, you like to breathe, and we breathe oxygen due to bacteria. Bacteria also are involved in all of the major nutrient cycles on the planet, including the nitrogen cycle. They make complex carbon structures, which allow for higher organisms to exist, such as plants and animals. And in general, they are just amazing little bugs. But how do they do all this? How do they do all of these mechanisms and pathways? Well, they have this informational code, which is hundreds of millions of years old, and it's been passed down from one cell to another. And this code is DNA. And I'm sure most of you have heard of DNA, but it's the informational string of life. And I like to kind of convey it to you as a string, especially for those non-biochemists and non-biologists in the room. So there's a string and it has 4 bases: adenine, thymine, cytosine, and guanine. And don't worry, I won't go too much more into them other than that. But these strings are bonded in a phosphosugar backbone. And 2 units of a phosphosugar backbone with their little bases come together and they form a secondary structure, the double helix. And I'm sure everybody knows the story of the double helix. So from the double helix, which is the secondary structure of DNA, you can come and form tertiary structures and quaternary structures and all these very complicated things. But essentially all this is a way to compact the large and oftentimes very dense code into a smaller structure. But I'm not going to go further into detail than that. So how does this information become functional in the cell? Like if it's in this code and you don't want to really perturb this code too much, how does the cell know what to do? How do things happen inside the cell and outside of the cell? Well, this information is transcribed in a process called transcription. Very simple to understand. So you want to transcribe because you want to maintain the integrity of the main informational book, the DNA. But you also want promiscuity in the message. You want the message to be able to do things. And these messages that are transcribed are known as RNA sequences or RNA strings. And they're comprised of our 3 familiar adenine, cytosine, guanine, but also now introducing uracil. Now you have tRNA, mRNA, which is messenger RNA, and rRNA, ribosomal RNA. And those are the 3 main gene products of RNA. But you also have other forms of RNA, such as small interfering RNA and small subunit RNA, but I'm not going to talk about any of those. They do other things. Now, when you're talking about coding, you're mainly talking about messenger RNA. And these are genes. So these are messages in a string that get translated into the catalytic, structural, and informational products inside the cell. And these are known as proteins. I'm sure you've all heard this word before. Proteins do all of the work inside, most of all of the work inside of the cell. They form, porins or channels in the membrane that allow molecules to flow inside and outside of the cell. They allow for structural integrity of the cell, so the cell just doesn't collapse on itself, especially in the case of eukaryotic cells. And they do the transformations of molecules inside that cell, so catalysis and metabolisms, breaking down sugar and forming complicated sugars. And these proteins are comprised of amino acids. Surprise, we're going to talk only about amino acids. No, I'm not going to go through all of these because it would take too long and nobody wants to sit here through that. But it's important to know that the bases code for these amino acids when you're talking about proteins. But how does this translation happen? Who is the monk sitting there going from ancient Latin into Old English? Well, that is the job of the ribosome. It is the major hinge of information inside the cell. So the ribosome is composed of that ribosomal RNA. So the ribosome is actually a complex superstructure that has some small subunits of ribosomal RNA and some large subunits of ribosomal RNA. And I'm sure you've heard the terms 16S maybe before, or 30S. That's confusing, and I'm not going to describe them in that way. I'm just going to say there's a small subunit, there's a large subunit, and they come together. And for some of us microbiologists out there, we also not only care about the structural and catalytic part of the ribosome, but also the taxonomic information that it codes, because if you ever look in taxonomy, oftentimes species are defined by their 16S, which is one of my favorite structures. So I have that pictured here, and it is E. coli 16S gene, and it's a pretty rainbow of secondary structure loops. And so you can see, just like DNA, RNA forms these secondary structures, and it's easy to imagine how these loops and turns can all fold in on themselves and form tertiary structures. And then when those subunits come together, they, like any A-list star, get adorned into this big complex with accessory proteins. And that's what all those chain bows are on top of the sprawled core of the ribosome. So you have your small subunit, your large subunit, and then these accessory proteins. And no, I'm not going to go into all of the accessory proteins because, yet again, it would take too much time, and honestly, I don't do that research. But isn't it pretty? So so what I just went through very briefly and very hopefully very simply, and didn't use too much jargon, is the central dogma of molecular biology. It's the central dogma of informational flow inside the cell. You have your main core of information, your DNA, and this is the book of life. And then this gets transcribed into short messages that go out and get translated and do things. So it is an oversimplification of how information flows inside the cell. It is by no means correct, but it's the easiest way to convey it to all of those learning this for the first time. Or maybe for the second time, but you haven't taken a biology class since high school. Okay, so that's fascinating. But, how did all this begin? So as you're going to hear today from some of the talks or see in some of the posters, amongst all of you, or the group is riddled with prebiotic or origins of life investigators. And what they're looking at is just what the name prebiotic implies. Before a large functional cell comes together, what happened? How did this information become more and more complex? Well, the predominant hypothesis or thought is that the origins of life actually started as an RNA world. So you have an RNA string that started and became more and more complicated with time. Now, this would imply like a ribosomal world. So you're not just talking about a string of information. You need that information to actually be functional too. And ribozymes, as I said, are catalytic as well as informational. So they're obviously the dream of all origins of life researchers. Obviously, I'm not one, so I'm talking in broad general terms for all of you. But what they're looking at is they're looking at the evolution of the ribozyme. So how has it changed through the 3 billion years of life on this planet? And also, how is it different, or how is it actually very similar amongst all the branches of life? So that's more of the evolutionary and the taxonomy and the phylogenetic. They're also looking at, some of them look at non-canonical bases, which, if some of you know Bradley Picard, that is what his research was as a PhD. student. So you're looking at, obviously, we have the 4 bases now for RNA, but was that always the case? Probably not. And there's actually a multitude of base units that can be used or incorporated into RNA. And so that would be non-canonical base research. And then there's others that also look at all of those blinged out, bedazzled accessory proteins on the interior, kind of middle area and exterior of the ribosome. And so that's looking at how these proteins actually modulate and influence the in general overall functionality of the ribosome. So now that I am done with my very simple and very quick talk, I talk a lot faster when I'm nervous, even if I'm looking at my own screen and not any of you. But now it's time for the real research. So I hope that some of the concepts that I briefly went over will allow you to understand their very awesome research, because I think that we have a pretty good conference this year and attendees. So with that, I thank all of you. I don't have anything else unless somebody wants to ask some questions, and I'll attempt to answer them. And Brett will be taking over as chair for this session since I am not there and I can't regulate stuff.
All right, well, let's thank Sam for that nice warm-up talk.